everyone, welcome to the course on computer aided drug design. Today we will talk about the PGB efflux and few more drug likeness property. Yesterday after blood brain barrier penetration I introduced what is PGB. We will spend more time on this PGB. They are P glycoproteins. These are efflux transporters, that means they throw drugs out from the cell inner, so that they maintain the concentrations of drug at very low values. They are meant to do the job for toxins and toxic metabolites. These are transmembrane glycoprotein, okay. They are 70 kilodalton in size. They are part of the ATP binding cassette family of transporters. There are 50 of them, large number of them, as you can see. These are cell membrane that pumps many foreign substance out of the cell. They are present in many tissues, in fact more in some of the tissues like a BBB. Like I mentioned, blood brain barrier has a lot of efflux transporters, small and large intestines, liver, kidney, adrenal gland, pregnant uterus. Increased intestinal expression. So if you have a large amount of PGB present in the intestine, so what it will do is it will reduce the absorption of the drug through the GI to the plasma, okay. So because it will try to efflux and remove the drug through the fecus. They are meant to remove toxic metabolites and xenobiotics. In fact, that is their job, to throw them into urine, bile and intestinal lumen, okay. That is the job, but unfortunately they may even throw the drugs as well. This picture was taken from this reference. So these removes hydrophobic substrates directly from the plasma membrane. So we have the plasma membrane just gets thrown out here, okay. So the ATP plays a very important role here, okay. That is what the P glycoprotein, they are on the membrane bound, as you can see here. Both the sites are essential for this. So for example, it's got two different sites, okay. So both are very important, and mutations in either site knocks out transport function. The site works sequentially, only one site at a time binds and hydrolyzes the ATP present here. So you require hydrolysis of two molecules of ATP to transport one molecule of this drug. So that is what it does, throwing out drugs away, and they are located at the membranes. So what is the physiologic role of this P glycoprotein? So we have oral intake, so we may have PGBs here, so it binds toxic material and then it's excreted, fecal excretion, okay. Or if it is in the intravenous, okay, then it binds and then it removes it through the urinary excretion. So these are the two main jobs of P glycoproteins, okay. So you have very hydrophilic material, it may go through the urine, hydrophobic, you may go through the fecal and so on. So there are large number of PGBs. These are the common names, MDR1, MRP1, MRP2 and so on here, as you can see. These are the systematic names. What are the substrates for these PGBs? Neutral and cationic organic compounds, okay. That is where your drugs may get caught. Then we have MRP1, conjugates organic anions, conjugates antifolates, bile acids, etoposide, okay. Nucleoside analogues, methotrexate, anionic cyclic pentapeptide. Where are they found? For example, these PGBs are found in intestine, liver, kidney, blood brain barrier. This is where we are talking about most of your drugs, neutral and cationic organic compounds. MRP1, they are found widespread. These are found in intestine, liver, kidney and so on. Actually, if you look at MRP5, widespread, and but they take in nucleoside analogues, cyclic nucleotides, organic anions and so on actually, okay. As you can see here where they are found, this is taken from this particular reference. So 50% of human cancers express large amount of P glycoprotein. So what it does, it throws the drugs out, so the drugs do not act, so it becomes resistant, okay. The cancer becomes resistant to drug therapy. Cancers which acquire expression of PGB following treatment of the patient include leukemias. That means after treatment, initially the treatment may appear to work, but as they started expressing lot of PGBs, then they start becoming resistant to those drugs. Cancers like leukemias, myeloma, lymphomas, breast cancer, ovarian cancer and so on, it happens. So, there are a lot of PGB antagonists that are currently either in clinical trials or about to enter the market. So, these are some structures as you can see, and these are new PGB antagonists. Okay. So, how do I design molecules if I want to avoid this PGB? Compounds with molecular weight 250 to 1850 are transported. So, most of the drugs, as you know, can fall into this. So, if you have N plus O greater than 8 or equal to 8 and molecular weight greater than 400, acid with pKa greater than 4, then they can become a PGB efflux, that substrate. That means they can get thrown out by PGB. If N plus O less than 4, molecular weight less than 400, acid with pKa less than 8, okay, then chances are they will not be PGB substrate. So, what it means is I try to keep my nitrogen and oxygen quite low. That means make it not too much hydrophilic in nature, make it like basic here. Reduce PGB efflux by introducing steric hindrance to hydrogen donating atoms. So, if you have OH, put OCH3, if you have NH, put NCH3, attach bulky groups, methylate the nitrogen. So, if you have NH, remove that H with CH3, decrease hydrogen bond acceptor position. That means add electron withdrawing group adjacent, add acid groups, change log P so that it does not penetrate into the lipid bilayer. So, these are some strategies for reducing PGB efflux. Okay. Nitrogen plus oxygen 4, 33% chance of efflux, nitrogen plus oxygen 6, 65%, nitrogen plus oxygen 8 or 9, 87%. So, hydrogen bond acceptors could be a big culprit for PGB. So, if you have large number of hydrogen bond acceptors, the risk of efflux is very high. So, like I mentioned, okay, try to reduce that by putting in some bulky groups nearby. Okay, so we talked about PGB. Let us again go back to the metabolisms in the ADME, and let us look at how to overcome these biotransformations using structural modifications. Now, once more to revise, we have two types of metabolism, phase 1 biotransformation and phase 2. Phase 1 generally it is oxidation, that means add O, okay, reduction, hydrolysis, okay, by esterases enzymes. Phase 2 is coupling these sites after phase 1 transformation by glucuronide with glucuronic acid or sulfate, like in this picture. No, you have an example of phase 1 hydroxylation, and then you are putting a SO sulfate has come here. So, it has become more hydrophilic, so it can go through the urine. Okay, so what can we do to avoid phase 1? So, what does phase 1 do? Okay, oxidation, reduction, hydrolysis. Phase 2 couples groups for existing. So, you can block sites by adding fluorine group. For example, this can become OH, okay, then it is easy to put the phase 2 SO3. So, what you do is, if you have a fluorine type of group, this type of reaction does not take place, understand. So, that is blocking metabolic sites by adding fluorine, that is a good strategy. Adding large groups, block metabolic sites by adding large groups, remove labile functional groups. Okay. So, as you can see, IC50 is 1.3 nanomole, stability is only 1%. Okay. Look at this. This is a delta type opioid receptor, IC50 0.3 nanomole, stability is only 4%. So, what has been done here? You put in some large groups here, okay, okay, and then changes have been made here in all these places. So, stability has increased to 52%. Okay, so metabolic sites, you add large groups, you also prevent labile groups like this leaving, okay, by having large groups here, and again you can see large groups here, okay. So, by doing that, the stability of biotransformation has come up quite a lot, as you can see here, okay, because if you remove this and make a smaller group, the stability comes down here, okay. What other things? Cyclize, that means instead of linear, make it cyclization, change ring size, change chirality, okay. Reduce lipophilicity, make it more hydrophilic. The metabolic enzymes have hydrophobic sites. Replace unstable groups. For example, if I replace the piperidine with piperazine, okay, so then this becomes more stable than this. So, what all can we do? We can cyclize, thereby it prevents the metabolism. We can make the ring size different, change chirality, make it more hydrophilic, okay. Okay, so the metabolic enzymes have hydrophobic sites, so biotransformation. So, you see, in some cases we say make it more hydrophilic, but if you want a drug to dissolve in the lipid layer and get absorbed, you want it more hydrophobic. If you want a PGB efflux preventing substrate, we do not want too much of nitrogen and oxygen. So, there are conflicting needs depending upon what we are looking at. So, remember that drug discovery drug design is not straightforward, okay. When you try to improve a parameter, another parameter can get disturbed, okay. Replace unstable groups. So, here we are trying to replace unstable groups here by replace as you can see piperidine with piperazine. Now, how do I prevent phase 2 metabolism? Introduce electron withdrawing groups or steric hindrance, okay. For example, you have this in the phase 2 metabolism, it can have like this. So, it makes it into a hydrophobic molecule. Imagine when I put a chlorine, this is an electron withdrawing group, this particular attachment does not happen, okay. Whereas, when I have this type of group, what happens? UDP glucuronyl transferase phase 2, it catalyzes the transfer of glucuronic acid component of UDP glucuronic acid to a small hydrophobic molecule. So, you end up glucuronic acid coming here. Whereas, if I have an electron withdrawing group like Cl, what happens? This does not take place, that means phase 2 will not happen. For example, you have a phenolic hydroxyl to cyclic urea, okay, that means a cyclization. So, the bioavailability as you can see increases from 0.6 to 87%, big change, okay. Change phenolic hydroxyl to cyclic urea or thiourea, that is what you are doing here, okay, cyclization. Change phenolic hydroxyl to prodrug. I did talk about what is a prodrug. Prodrug, it's taken in orally, it is not a drug, it may contain another component which get cleaved in the plasma region because of the action of some esterase or lipase, and one part will be the real drug which will start acting. For example, if you look at this particular drug, terbutaline, it is given 3 times a day because it has got very poor stability, okay. Whereas, you make a prodrug out of it, bambuterol, okay, you put in these 2 groups, as you can see these are ester bonds which can get cleaved with the esterase enzyme. So, these are called long acting beta adrenoceptor agonist used in the treatment of asthma. It is given only once a day, okay. Whereas, this has to be given 3 times a day, whereas when I put in this 2 groups here and here, it acts as a prodrug, it is stable, but it cleaves only in the presence of esterase type of end because you have the ester bond. So, it is given only once a day, it is used in the treatment of asthma, you can see that. Chirality affects metabolic stability, okay. Enantiomers bind differently with the metabolizing enzyme, right. You have R enantiomer, S enantiomer, and they may bind with the enzymes which are involved in the phase 1 and phase 2. So, you may end up with either biotransformation happening or not happening. Okay, next one is plasma stability. Compounds decompose in plasma due to the presence of hydrolytic enzymes. Plasma contains hydrolytic enzymes, so ester bonds can get degraded. Plasma stability increase with steric hindrance, so you do not allow the hydrolysis taking place, or you can put electron withdrawing groups or replacement with less reactive groups. Okay. For example, look at this compound fluocortine butyl, it treat or prevent skin disorders. Okay. So you can have this bond breaking because of the hydrolysis. Okay. Esters can get hydrolyzed, amides, carbamates, lactam, lactone, sulfonamide. So if you have these groups, they have chances of poor plasma stability because there can be hydrolysis taking place here, as you can see here. Okay. This bond can break. So what we can do, we can have big groups like here you can have electron withdrawing groups, so this cleavage does not happen. You can replace with less reactive groups so that the cleavage does not happen. But remember, these type of groups can have plasma instability. Esters, amides, in this order, esters are extremely susceptible to hydrolytic enzymes, amides, carbamates, lactam, lactone, sulfonamide, and so on actually. So this is the strategy by which we can enhance plasma stability. Okay. So what are various things we can do? Replace esters with amide, because amides are less hydrolyzable than esters. Increase steric hindrance, add electron withdrawing groups, eliminate hydrolyzable groups. Okay, these are the various strategies, okay, we can do to improve plasma stability. Of course, we also have these proteins in the blood plasma, and compounds can bind to that, thereby their amount, their concentration in the freely available form will be very less. So we want to avoid that also. So blood plasma proteins are albumin, like human serum albumin, alpha acid glycoprotein, lipoprotein. These are the proteins found in the blood plasma. So organic anion, carboxylic acids, phenols bind strongly to you. So if you have a acidic drug, highly acidic drug, chances are it can have a high plasma binding capability. So the amount of freely available drug in the blood will be reduced because of this. Amines, basic drugs and hydrophobic compounds like steroids, they bind to this glycoprotein. Okay. Whereas acid drugs can bind to albumin. So what does plasma protein binding effect of that? It affects the ADME of the drug, it retains drug in the plasma compartment, restricts distribution of drug in the target compartment. That means because some of the drug is bound to the plasma, so the amount of drug available at the target site may be low. Okay. Then decreases metabolism, clearance, and it prolongs the half life, because some drug is already bound to the plasma and it may get slowly released over a long period of time. So you may have a fast drug available, whichever is in the free form, whereas the drug that is bound to plasma will get slowly released, and concentration of the drug will keep persisting. That is why it prolongs the T half. It limits also the brain penetration, because the amount of drug available is very less. You need higher dose loading, but reduces maintenance loading. So although you require higher doses, because some drug has gone to the plasma bound, the drug available for maintenance has gone down, but you require higher doses of loading. Okay, for example, one example, MK886, it is a drug, it is an acidic drug. It has been found very good for enzymes like prostaglandin E synthase, MPGS1, which is involved in arachidonic acid inflammatory, but because of the high plasma binding, the drug has to be withdrawn. So it is an acidic drug. So what are the structural modifications we can do? Reduce lipophilicity, so log P for acids and log D for non-acids. Reduce acidity, okay, increase pKa of acid, increase basicity, increase pKa of the base, reduce non-polar groups, non-polar area, okay, or non-polar groups as we can see, okay, that is called a reduce non-polar groups, reduce non-polar area, increase polar surface area, that means increase polar surface area but reduce non-polar surface area, increase hydrogen bonding okay, and so on. So many different strategies one can follow to reduce the plasma protein binding effect. Okay. Toxicity is another important parameter, the drug toxicity which we need to consider. So when we say ADME, now it has been extended to ADMET, absorption, distribution, metabolism, excretion and toxicity. So the concentration of the drug, the actual concentration that is used for treatment should be nowhere near its toxic limits. The toxicity limits should be much farther. Okay. For example, if this is the activity, efficacy, okay, for example antibacterial, if you take, as I keep increasing concentration it starts killing bacteria and it rises and reaches plateau, but the toxic limit of that drug should be much far. Okay. So I can operate in this concentration range but still it will not be toxic, whereas if this graph is closer then this range becomes narrower. So I can operate with confidence because the toxic limits comes only beyond this, do you understand? Okay, the toxic limit is only beyond this, whereas this is the efficacy limit. So ideally I should have a drug which shows a graph for the activity and which shows a graph for the toxicity. Whereas if I have a graph for a drug which shows like this, activity like this, toxicity, if I start using the drug in this concentration it can also have some toxicity as well, okay, which is not allowed. Whereas if I start using the drug at lower and lower, the efficacy of the drug becomes lower and lower. So if I want to prevent toxicity then efficacy is low, if I increase and improve efficacy then it also has toxicity. So ideally we should be having drugs like this where the toxicity graph is far away from the activity graph. So this is the very ideal situation, but many times you can have toxicity coming in here like some of the chemotherapy drugs and so on, which can be also toxic and it is active because you have to give the drug at very high concentration. So we need to understand toxicity as well. The toxicity could be short term, long term, toxicity could be systemic, toxicity could be particular organ, different types of toxicities are there, we will look at some of them in brief. Okay. The reactive metabolite, so that could be toxic, gene induction, mutagenicity, oxidative stress, autoimmune response. So these are the various ways by which toxicity could be introduced because of the drug treatment. So what are the substrates that may initiate toxicity? Amines play a very important role, aromatic amines, hydroxylamines, nitro compounds. So generally nitrogen based compounds can lead to toxicity. So you can see aromatic amines, hydroxylamines, aromatic nitro, nitroso, okay. Then comes alkyl halides, polycyclic aromatic, alpha beta unsaturated aldehydes, nitrogen containing aromatic, bromine aromatic, thiophene, hydrazines, hydroquinones, azo, vinyl, phenolic, aliphatic amines, furans, pyroles, imidazole, medium chain fatty acids. So it comes like this. So these type of structures, as you can see, many of these structures can be toxic because some of the metabolites could end up being toxic, okay, that is a big problem, okay, and the toxicity can be of different in nature. Acute toxicity from single dose, I give one dose and it is toxic, so that is called acute. Chronic is from long term dosing, that means the drug is given over a period of few weeks, few months, few years and so on. Cytotoxicity, so the drug kills cells, that is cytotoxicity. Genotoxicity, there is some mutagenic change to the DNA. Immunotoxicity, the immune response is affected, okay, immune response is compromised that is called immunotoxicity. Teratogenicity, embryotoxicity. Okay, so different types of toxicities. So one needs to study all these if one is designing drugs, because it might not be a single dose, it could be long term dose, cell death, lot of mutagenic changes to the DNA can happen, it may affect immune response. So some of these toxicity you may be able to find out on long term studies, some of these could be the short term, and some of them needs to be studied in very specialized condition to look specifically looking at embryo and so on actually. In fact, there is a website http Coffer Informatic, you look at this QSAR web service that predicts toxicity properties and provides fragments to aid interpreting predictions. Okay, so you give a structure, it may able to predict the toxicity of that compound and also look at the fragments and also make comparison and certain conclusions on. So there are many websites and this is quite a good website. When we have time we will look at this as well. Okay, so we looked at toxicity, okay, that is another aspect. Now let us look at another important aspect, that is called HERG, the human ether-go-go related gene. Okay, human ether-go-go related gene. This is a gene that codes for a protein known as Kv11.1, the alpha subunit of a potassium ion channel. This ion channel contributes to the electrical activity of the heart that coordinates the heart beating. So this particular, we have to look at this particular gene and see whether the drug is going to affect this, because if so it may affect the heart beating. Okay. When this channel's ability to conduct electrical current across the cell membrane is inhibited, it can result in a potentially fatal disorder called the long QT syndrome. So as you know, if you remember how a typical ECG looks like, so we have the initial P, okay, and then we have this is called the PR, then goes down Q, then R is high, and then again it goes down S, then this is called T. Okay, so sometimes this gets extended, okay, that is called the extended QT syndrome, okay, long QT syndrome. Okay, so because the potassium ion channel gets blocked because of your drug, and this can lead to cardiac problem. So FDA suggests the establishment of a cardiac safety profile during preclinical drug development. So you need to study whether your drug affects this pattern, especially the Q and T part of it, QT. This is called the QT interval, okay. This is called the PR segment, this is called the ST segment. So whether there is a drug affects this is what they need to see. So we need to look at this. So generally drugs are checked to see whether it affects this particular potassium channel and hence the QT part of the time. Look at this drug, Substrates, symptomatic treatment of adult patients with nocturnal heartburn due to gastroesophageal reflux disease. Okay, so as you can see, sometimes you can have acid reflux coming in, then it can lead to heartburn. This drug was withdrawn or had its indication limited due to the report about long QT syndrome. So this drug elongates the QT time period. Okay, another drug, terfenadine. This is allergic rhinitis, hay fever, allergic skin disorders. This was withdrawn due to the risk of cardiac arrhythmia caused by QT interval prolongation. Again, it's because of long QT interval. So these drugs directly affect the cardiac functions. Okay, so they were withdrawn. So you need to understand whether the potassium channel ion channel gets affected because of blocking of this HERG gene, which in turn affects the cardiac rhythm, especially the QT portion of the ECG. Okay. So, what are the structural features that lead to binding of the drug to HERG? Basic amines: positively ionizable pKa greater than 7.3, log P greater than 3.7, absence of negatively ionizable groups, there are no negatively ionized group, absence of oxygen hydrogen bond acceptors. So, these can lead to so what modifications we can do to prevent HERG binding? Reduce pKa, okay, reduce pKa, basicity of amine, okay. So, there is a mistake here, reduce of amines, reduce pKa of amine, make it basic, reduce log P, okay, because add acid moiety, add oxygen hydrogen bond acceptor, because if it is absence then chances are it could be binding to HERG channel. Rigidify linkers, that means reduce flexibility of these molecules. So, by doing that we can prevent this HERG binding, and hence we can prevent the disturbance caused to the potassium channel, and hence the cardiovascular arrhythmia, extended QT syndrome. Okay. So, we will continue more on this drug likeness in the next class as well. Okay. Thank you very much for your time.